example, if you have an individual with type A blood, if you were to add serum with anti-B antibodies, this will not cause clumping of cells, as anti-B antibodies will not bind to the surface of these red blood cells. However, if you bind anti-A antibodies to A blood, it will cause their clumping. And using this technique, which is further expanded upon in figure A7 in your textbook, you can use this to identify the blood type of patients or animal models. So now that we've gone through a couple means of diagnostic testing by using monoclonal antibodies, let's go through a visualization technique that is used fairly often in the lab, which is microscopy and imaging. So microscopy and imaging is based on the idea that you can have monoclonal antibodies against a very specific surface protein for a cell, and these monoclonal antibodies are labeled with a fluorophore that is excited at a very specific wavelength and then can be detected by microscopy. So this technique is also called immunofluorescence microscopy. So the antibody will stay very stably bound to its antigen, and you can remove any unbound antigen from this from your sample. So for example, here we're looking at islets where these scientists have used antibodies that are specific for beta cells of the pancreatic islets, and these antibodies that are specific for the beta cells were labeled with a green fluorophore. So you can see these patches here of green that contain your beta cells. Now your alpha cells were labeled with an antibody specific for your alpha cells. However, they don't have a green fluorophore, instead they have an orange fluorophore. So you can see these patches here that contain just your alpha cells, and then you see these populations of alpha cells within your beta cells. So you can see that this is a really important technique that allows scientists to specifically look at tissues to see the cellular composition of these tissues. Now let's go to more types of ways to identify cells within a solution or within a tissue. So we're going to be talking about flow cytometry and FACS analysis, which allows you to identify specific cell types in a sample. Now this technique is based on the idea that different cells within your immune system or within tissues have different surface markers. So surface markers are proteins that are expressed on the outside of a cell, and different cells have different variations of such surface markers. So if you were to take a mixture of cells and you want to see the different compositions of the cells within your solution, or you would like to isolate a specific cell type, you can take cells or your mixture of cells and add into this mixture a variety of antibodies that are specific for specific surface markers, and each of these antibodies will be labeled with a different fluorophore or a different colored protein. So for example here, we have these antibodies that are labeled in red that are specific for this red surface molecule here, and then the antibodies that have a green fluorophore that are specific for this blue triangle surface marker. Now, the way that flow cytometry works is that it's as each cell passes through a laser beam, seen here, it scatters the laser light of these fluorophores here and the dye molecules, and this gets the fluorophores excited, and subsequently they fluoresce. Then the machine can detect which cells are fluorescing based on the laser that came at them, and you can identify what antibodies are bound to which specific cells, and thus you could know what surface molecules these cells have. So in this example here, we're looking at surface staining of IgM and IgD. So these are two immunoglobulin subtypes. And you can see here, this is the labeling of IgM on the x-axis on this right side, and IgD on the y-axis on this right side. And each of these dots in this plot represents one cell. So, for example, this dot right here has a lot of surface IgM, as the as the distance along the x-axis determines the intensity of the fluorescence from the cell, such that the more IgM molecules this cell has on its surface, the more IgM-specific antibodies will bind to the cell, and these antibodies subsequently will be excited, leading to higher fluorescence. So this cell has very high levels of IgM. However, you can see that it doesn't have high levels of IgD. Another couple dots, let's go through some examples. For example, this dot here has low IgM but high IgD. So this cell has low levels of IgM compared to this cell here, but it has much higher levels of IgD. And you can use variations of these plots. So for example, dot plots is where each dot represents one specific cell, or you can use counter maps, which looks at the density of cells within a particular area to understand the composition of your sample. So you can see here that a majority of this population within the sample is IgM, both IgM and IgD positive. They're in this quadrant, or they're IgM, IgD double negative cells. Now on the left, what you're seeing is a histogram. So this takes the entire population of cells and sees how highly expressed each specific surface marker is. So the x-axes are the same relative to these plots here. However, on your y-axis, you have a cell count. So for example, with IgD, you can see that this low population here, there's a fair amount of cells that express low amounts of IgD, and there's a lot more cells that express high IgD. Now these cells can either just be analyzed just like this, so you can look at a sample and say what populations of cells you have. You can also use flow and FACS to separate out cells so you can isolate a specific population for further assessment. So FACS is really great. So cell sorting is really great for isolating a small number of cells in pure form. However, mechanical means of separating cells is preferable when you need a large amount of cells to work with for your experiment, and this can be prepared very, very quickly. So this technique also uses monoclonal antibodies. However, instead of being labeled with a fluorophore, these monoclonal antibodies are coupled to paramagnetic particles or beads. So in this example here, the experimenter would like to isolate the cells that have this red circular epitope on their surface. So what you could do is you could take your heterogeneous population of cells and mix them together with antibodies that are coupled to paramagnetic beads that are specific for the surface antigen you're looking for, in this case, the red epitope. So you mix these cells and the antibodies will bind to their specific epitopes, and then you can pour this mixture onto an iron wool mesh such that the magnets, when you put a magnetic field onto this iron wool mesh, the cells that are coupled to the paramagnetic beads will then remain stuck within the solution, within the iron mesh when you flow through a PBS or any other solution that you're using, such that the cells that you would like remain within the iron mesh because they are coupled to magnets and you have applied a magnetic field to your wool mesh, and the cells that you don't want will be flushed away from the wool mesh. Then, when you release or remove the magnetic field, the cells that are coupled to your antibody will then be released. So now you have a population of cells that does not contain the epitope that you are looking for, and a population that does. These cells can then be used in further experiments. So now that we've gone through how to identify cells and isolate different types of cells using monoclonal antibodies, let's go through a couple of antibody-based tests to test for cellular functionality. Now, the first technique that we'll be talking about is called intracellular staining. So, as you've heard in a variety of lectures, B cells and T cells and other immune cells, when they're activated, will produce certain cytokines or certain proteins that signifies that they are activated. So, for example, here, this T cell is producing activation markers within the ER. So as cells are producing these activation markers, you can stain them intracellularly, meaning within the cell, to see how many cells within your population are activated or producing the cytokine of your choosing. So in this example, you take activated T cells and you treat them with an inhibitor that blocks protein export. Now this is important because you want to make sure that all the proteins that you'll be staining for remain within the cell so that you're not losing any signal. So these cytokines will subsequently remain within the ER. Then you fix the cell, which means you kind of freeze it within the state that it's in, and you permeabilize it with mild detergents, such that this creates pores within the membranes of this protein. And through these pores, you can put in antibodies that are specific for the cytokine of your choice. Now, these antibodies are labeled with fluorophores, as you saw in the flow cytometry and FACS method, and you can subsequently run these cells on a flow cytometer and also look for fluorescence, very similarly to flow cytometry, but just staining intracellular proteins as opposed to cell surface proteins. Our next technique is called cytokine capture. So in this technique, it does not depend on preventing proteins from leaving the cell. In fact, you do want the proteins to be leaving the cell, because the way that this works is a hybrid antibody is made that is specific for both the T cell receptor or an MHC class I marker, as well as the cytokine of your choosing. So as opposed to typical antibodies, where both chains are specific for the same epitope, you're generating an antibody that's specific for both a cell surface marker of your cell and the cytokine that you're looking for. So this technique is really good to see whether the cells that you're specifically interested in are producing the cytokine that you're specifically interested in. So here, for example, this hybrid antibody is one for MHC class I on the cell surface of your cell and specific for these green cytokines that you're looking for. So if you take your population of activated T cells and incubate them with these hybrid antibodies, they will bind to your cell surface marker. Then, if you stimulate your T cells, and if the T cells are secreting the cytokine that you're interested in, the secreted proteins will then also bind to the antibodies that are already on the surface of the cell. Then, you can detect the presence of this specific protein within your sample by washing away any other proteins that may be secreted, such that antibodies specific for your antigen that's bound to your hybrid antibody will be there, and you can look for their presence by fluorescence or another chemical means. Now, let's talk very briefly about a technique to deplete specific cell types within the body. So you can, in both experimental animals or in the clinic, you can take monoclonal antibodies against a surface marker of a cell that you would like depleted and inject these into the patient or the animal. And then the antibodies will bind to the cells that contain the surface marker. And then with phagocytosis and other techniques and other immune-mediated mechanisms, these cells will then be depleted. This is important in such clinical cases as treating autoimmunity. Now, we've gone through a variety of different techniques that center around the use of monoclonal antibodies. And I want to emphasize the overall message to you that monoclonal antibodies target a specific protein, and they can be used to identify that protein either within a sample, within a tissue, or they can identify specific cell types. They can also be used to test functionality and to deplete specific cell types. So using monoclonal antibodies in immunology is really critical. Now let's move on to a couple techniques used to study the function of immune cell responses. So we'll be going through techniques of both ways to test CD4 and CD8 T cell functionality. And it's important to know whether or not your CD4 or CD8 T cells are functioning for a variety of reasons that we'll be going through. Now, the first experimental technique that we'll be going through can be used for both CD4 and CD8 T cells. A specific one of the signs of cellular lymphocyte activation specifically is lymphocyte proliferation. So once a T cell encounters its cognate antigen, these cells will be activated and they'll start proliferating very quickly. So testing for lymphocyte proliferation is one means to test for whether or not cells are activated in the presence of a given antigen. One way to do this is to label your cells with a protein called carbofluorescent cyclamidyl ester, or CFSE, which is much easier to pronounce. So you can take a population of cells and label them with CFSE. CFSE will enter the cell, and once it's within the cytosol, it will become covalently coupled to the lysine residues within cellular proteins. Now, once you label your cells, this is considered 100% labeled. And then, because the CFSE is within the cytosol of these cells, every time your cell divides, it will lose half its fluorescence intensity, such that after one cell division, your cells will have 50% of your CFSE, and after another cellular division, they'll have 25, etc., etc. And you can actually then use FACS, or flow cytometry, to see the level of CFSE. So for example, in this plot over here, this 0 signifies your 100% fluorescence intensity, and then you can see that there's the next bump over is about half of that mean fluorescence intensity, such that this signifies cells that have undergone one cellular division. This peak, again, is shifted down, signifying even less CFSE staining, signifying 2 cell divisions, and so on. And this technique works really well for looking at cells that have divided 6 or 7 times. So it's a great mechanism to look for lymphocyte proliferation. Now let's shift over into using techniques that you can use for studying the activation of CD4 T cells. Our first technique that we'll be looking at is antigen-specific T cell proliferation. So in this experiment, you take a mouse or any other experimental animal and immunize them with antigen A. So this is your antigen of choice. Then, after about 5 to 10 days, so this is about the time by which your adaptive immune response will be activated, you can remove cells from the lymph node of your immunized animal, and then you can capture these cells, Then you can take the cells that you have isolated from the lymph node of your experimental animal and culture them with antigens A or B or whatever else. So you would expect that in the presence of antigen A, the antigen that you immunized your experimental animal with, there will be a lot of cellular proliferation because your activated cells will be coming into contact with the epitope that they are specific for. And if you were to culture them with an antigen for which your experimental animal was not immunized for, there will be no proliferation. So then you can look at proliferation just by cell density or cell count, and you'll see that for antigen A, you'll have a high level response. So this is a great technique to see whether or not your experimental animals can respond to a specific antigen. So if you want to study whether animals with different genotypes or with different proteins intact or not will still respond to an antigen, this is a great technique for CD4 T cells. And we'll get into animals with depleted or added genotypes in a couple slides. Now next we can cover techniques to study immune cell responses of CD8 T cells. So the first experiment we'll be talking about is target cell killing. So activated CD8 T cells can generally kill any cell that displays the specific peptide MHC class I complex they recognize. So the simplest and quickest bioassay to see whether or not a population of CD8 T cells is capable of killing its target cells is by just looking to see whether they'll physically kill these cells. So what you do in this technique, which can also be called 51 CR release assay, is you take live cells that'll take up but do not release radioactively labeled sodium chromate. So you label your target cells with sodium chromate, and then you add cytotoxic T cells that you think will kill these particular target cells. So what's important is that these live cells can take up sodium chromate, but they cannot release them. The only cells that can release the sodium chromate are those that are dying. So if your cytotoxic T cells can kill your labeled target cells, this will lead to the release of radioactive chromium. However, if your cytotoxic T cells are unable to kill your target cell, this will not lead to the release of the radioactive chromium. So now we'll be going over an assay to look for the presence of cytotoxic T cells within experimental animals, or in vivo. So this experimental technique is based on the idea that you can differentially label cells with an intracellular dye, such as CFSC that we discussed earlier. So if you can put different amounts of dye into different types of cells and then differentiate these populations using flow cytometry. Now in this experiment, for this experiment, scientists were looking to see whether or not certain animals can produce a cytotoxic T cell response against a specific virus. So these different animals were vaccinated or immunized with this specific virus, and then multiple days later, so after a T cell adaptive response could be induced, different cells were injected into the mouse. The first population of cells was coated with the virus peptide that these mice were immunized to previously. So in theory, if there is a cytotoxic T cell response that this mouse is able to launch, these virus peptide-coated target cells will be killed by the cytotoxic T cells. In addition, into these mice were injected control cells that the cytotoxic T cells that were activated during the immunization, these will not be killed. So you can see here that within an uninfected mouse, you have equal levels of control cells and target cells. And these are differentiated, again, by CFSC staining. Then, if you looked at an infected mouse, these mice have the same level of control cells, again, because your cytotoxic T cells are not killing your control cells, but significantly lower levels of your target cells, signifying that this experimental animal indeed did launch an adaptive immune response against the virus that it was immunized by.